Okay. Of course that won't work. Okay. So last time we left off, just a bit of a review, and then we'll get back into things. We left off with like lipid structures, so primarily like triglycerides or fatty acids. It's mostly what I wanted you to know about, like 3 fatty acids and a glycerol molecule. Just knowing that that's what a triglyceride is, and that we store lipids and fats in essentially 2 places: one fat cells, another one intramuscular triglycerides. I would know that. That's going to come up quite a bit in exercise metabolism. And we were talking a little bit about the different types of fatty acids, and the main reason for this is the amount of carbons in them more or less dictates how much energy that we're going to get out of it. So I wanted you to get an appreciation for essentially how they're not all the same, how we can get different amounts of energy out of all of them, and how like they're somewhat different. There's a particular thing that you don't have in your notes, so I'm going to want you to like memorize here in a bit. But like overall, just looking at this, like 18, that's the amount of carbons in it. And then that 0 is what? What does that notate? Does anybody remember? Yeah, number of double bonds. Yes. So right here, both of these are saturated fatty acids, meaning that they have 0 double bonds in them. Right here, if it has 1 double bond, what does that mean? Evidently nothing. Okay, so what's up, Josh? Yeah, so it's unsaturated. So like broadly in terms of fats, we have 2 different camps. We have saturated, which have 0 double bonds, and like unsaturated, which have either 1 or many double carbon bonds. So this singular one means MUFA, or monounsaturated. So like everyone good with that? Mono, singular, like how I speak, monotone, right? Hopefully everyone understands that. Like moving on a little bit, like polyunsaturated fats. Now here, that 3, that notates that there's 3, and then that omega-3 means where the first double bond is on the 3rd, like carbon bond there. So hopefully you recall that somewhat. And we were talking a little bit about medium chain, like triglycerides or medium chain fats, and like them ranging somewhere between like 6 and 12 carbons. Now on the board I wrote like 8 to 12 because I can't really remember what the 6 carbon one is. So like it's not really super important for us to know, I don't think. But 8, 10, and then there's a 12 carbon one called lauric acid, which I wanted you to know because there's a little bit of, whenever they're studied, normally the lauric acid is the one that's actually looked at. Pretty interesting stuff, I think. At least I want you to know about that. So like then here is my question. Whenever nutritionists, dietitians, whenever we talk about fats, are they all equal? And like you don't really have this slide, but because there's not too much that I want you to understand from it. So hopefully with understanding the different structures of these, you get that they're not all equal. And a second ago, I told you that the number of carbons in it really dictates how much energy that we can get out of it. And there's a couple of other things within the structures where we can only make hormones out of certain ones of them. Like they're fairly complex, and like I don't know. Like I just want y'all to get the same appreciation for fats that I have, right? So like one thing right here. One thing that bothers me a lot whenever people talk, like I teach a nutrition class here too, people will talk about certain things being good for you or bad for you. So like saturated fats, who's heard that they're bad for you? Right, most people in here. Now it's more confusing than that because there's actually just about no relationship between the amount of saturated fats people consume and heart disease or stroke or anything like that. And sorry, I'm a little excited today. This is crazy. It's probably because I'm talking about saturated fats. You know what? Like that's actually true. There's always studies swaying back and forth. One study will be like, oh, it's going to kill you. Another study, oh, it's not going to kill you. So I'm not saying that saturated fats are not exactly bad for you. I'm saying at the very least it's murky, that like it's somewhat confusing. And here's something. Go and write this down. Amount of dietary saturated fat consumed has virtually no relationship with heart disease. However, this is where it's confusing. Amount of circulating saturated fats in your blood. Like I take your blood out, and I see the amount of saturated fats in your blood. That is associated with heart disease. Right? Now, do y'all see how that's confusing? Now, there's a couple of different ways how we can actually increase the level of saturated fats in our blood, but the main one is by not burning it. And hopefully everyone recalls what glucose looks like, C6H12O6. Is that a more or less simple molecule than some of these fats? What do y'all think? Yeah, like DJ's saying, more simple. I would agree with that. Our body typically burns the more simple thing and doesn't quite know what to do with a more complex thing. So overall, fats are typically more complex than carbohydrates. So part of the reason how saturated fat is unfairly maligned is individuals will consume carbohydrates or glucose or whatever and saturated fats. And the carbohydrate will be burned first, and then we will have a higher level of circulating saturated fats. And that essentially is just a proxy for people consuming too much energy. So I think all of us can agree that eating too much is bad for you, right? Unless you're trying to build muscle mass or something. But that's overall what that means. Okay, so I'm going to quit talking about that because this isn't really the class for that. I want you to understand the different oxidation rates of these fatty acids, and the length and number of double carbon bonds is actually relatively meaningful. And we've known this since 1986 in rats. So a couple of things here. Of the saturated fats, lauric acid, the one I told you, remember, the 12 carbon one, was most efficient energy substrate. The longer the chain length of saturated fatty acids, the slower the rate of oxidation. So I want you to know that, right? And that number 3 point right there, I think, is important for us to know. Oleic acid, or the olive oil fat, was oxidized at a remarkably fast rate, similar to that of lauric acid. I'll circle back to that in a bit. It seems like we almost evolved to burn monounsaturated fats really well. Of the omega-6 essential fatty acids, linoleic acid, the 18 carbon, 2 double bonds, omega-6, was oxidized at a faster rate than any of its metabolites, with arachidonic acid being oxidized at the slowest rate. So first thing that I want you to think about is this isn't rats, so who cares? This was done in humans in 2000, so they did some carbon, radio labeling, pretty interesting stuff. And we can see here the oxidation rates of these different things. So if we look 9 hours later, essentially this means the amount that is recovered in the breath, indicating that it was actually burned for energy or fuel. So that 12 carbon one, that's lauric acid. Hopefully everyone is good with that. The shorter the amount of carbons, it's burned really rapidly. And going on beyond that, it's 18-3, that's the omega-3 fatty acid. Alpha-linolenic acid, the only essential omega-3 that we find in flax seeds and chia seeds and so forth, right? And then it kind of goes on from there. Now, here is really the part that I want you to know. So oxidation of saturated fatty acids decreased with increasing carbon length. So here, laurate or lauric acid, that's 12 carbons. Palmitate or palmitic acid, that's 16 carbons. Stearate or stearic acid, that's 18 carbons. So they're essentially, what I want you to understand about saturated fats is what indicates how quickly you will oxidize them or burn them for fuel is how many hydrocarbon bonds are within it. Does that make sense? So here, it's following what I said a second ago in regard to glucose. Laurate, or lauric acid, is a simpler molecule than palmitic acid because it's less carbons, right? Now, here soon, this will have a tie-in for exercise performance, but not quite yet. We have to cover a couple things first. Now, in terms of unsaturated fats, here's what I want you to know in regard to this. They're in this following order. Linolenic acid, that's the omega-3 fatty acid. 18 carbon. Oleate, which is that's the monounsaturated fat. And linoleic acid, which that's the omega-6 fatty acid. Now, this next line is really what I want you to know. Oxidation of the 18 carbon, or just say unsaturated fats, was significantly correlated with the number of double bonds. So, big picture here, saturated fats are burned in the order of carbon length. Unsaturated fats are burned in the order of double carbon bonds. So the more amount of carbon bonds, it's burned faster. Now, why would that be? Has anyone ever heard of fats going rancid? Like if you have a cooking oil or something like that, then you smell it and it's gone bad? So that happens whenever a double carbon bond gets oxidized from a free radical or some reactive oxygen species or something like that. So in a way, the more double carbon bonds, which are the only points where a fatty acid can actually get damaged by light or oxygen or the air or what have you, it's more of an unstable molecule. So what I want you to understand here, and I know I'm saying that a lot because, well, I don't have anything better to say. Saturated fats burned in the order of carbon length, and unsaturated fats are burned in the order of instability. So if a molecule is unstable, meaning it can go rancid and bad stuff within your body will preferentially burn that first so that it doesn't have time to stick around and do anything bad. Is everyone good with this? I know it's relatively complex, but it's I think it's important for us to know and understand. Any questions about fats? I'm going to circle back to fatty acid translocase and some other stuff here in a bit. But any questions really quick? Yes, Eddie.
So saturated is burned in the order of the chain?
Yes. Yeah. Amount of carbon bonds. Yes. So order of the chain. Yeah, I'd accept that. Good. Okay. Moving on. So now let's talk about proteins a little bit. Obviously they're composed of amino acids, the building blocks of life, all of that type of stuff. Here's a term that I want you to know, gluconeogenesis. So some amino acids, not proteins, some amino acids can be converted to glucose, and these are called gluconeogenic amino acids. Now, I'm not going to make you memorize which ones they are, but just know that some do that. There's other amino acids, and write this down, that are ketogenic amino acids. So if anybody's ever heard about ketones, I'm going to explain to you what those are. But amino acids are generally in two camps, gluconeogenic or ketogenic. And that can have some consequences for overall metabolism. And they can be, they can be converted to various intermediates within metabolism, particularly the Krebs cycle, and I'm going to show you where that's happening because it's, I don't know, I think it's really cool how it's incorporated. But overall, protein is not a primary energy source during exercise. So on average, it's somewhere between 2 and 15% of energy during exercise. Now, there's a particular type of exercise that burns more protein, and other types of exercise that burn less protein. Can anyone guess what type of exercise burns the most protein? What did you say, DJ? Strength exercises. That's what DJ says. And I wanted him to say that. What are you going to say, Alec? Long distance running. It's almost like we've talked about this before, right? Okay, cool. So actually, the type of exercise that burns the most protein is long endurance events. Because, because we can only derive energy from it in an aerobic fashion. We can't do it anaerobically. Now, granted, I understand what DJ's thinking. it's really important for recovery after exercise, but during exercise, it has virtually no contribution to energy outcomes there. Now, here's the amino acid structure. There's an amino group, hydrogen group, carboxyl, and this variant group. Now, in order to make this work, we have to take off this amino group because that has nitrogen in it. And everything we've looked at or talked about so far really only has carbon, hydrogen, and oxygen. So like fats, that's what they have, that's what carbohydrates have. So that's really the odd thing out that we need to take off. And I want you to know what that's called. That is called deamination. And just spell it well. I know I don't say it well. And here, I've talked about this a couple of times. In regard to like muscle metabolism, there's relative certainty that the muscle can break down like about 7 or so amino acids, but 3 of them are probably like the most important. So valine, leucine, and isoleucine. Other ones that can break down are like, what is it? Alanine, asparagine, and glutamine, and glutamic acid, and arginine. But that's neither here nor there. Now, those 3 amino acids, I want you to know what they're called. Does anybody remember what they're called? I've said it before in class. Yes, DJ. Yes, branch chain amino acids. So go and write that down. BCAA. Branch chain amino acids. It seems like those are the ones that are oxidized to the greatest extent whenever we're exercising. And through the research, it seems like individuals doing like really long-term endurance performance, like I'm talking marathons, ultra marathons. What's his name? I can't remember. It seems like if you give them like supplements that actually have the branched chain amino acids in them, it increases their like capacity for endurance or makes them exercise just a little bit harder. So like it's fairly cool stuff, at least in my eyes. Any questions about proteins and stuff? Now, kind of back to the standard programming on bioenergetics, we're going to talk about ATP and like how glycolysis works and things. So let's go ahead and get going with that. Any questions? No? Okay, good. Okay, so high energy phosphates, ATP. This is the energy currency of the body. We need to know about this. It consists of adenine, ribose, which is just a 5-carbon sugar, but I'm not going to ask you to define this. What's really important is the 3-link phosphates, which on the next slide is going to come up, and these 2 equations overall. So synthesis. Hopefully everybody knows that how we make ATP is by combining ADP and an inorganic phosphate. Now that inorganic phosphate can come from numerous different places, but overall that's just the synthesis of it. Now the breakdown, ATP, ATPase being the enzyme that kind of cleaves it apart, yields ADP, inorganic phosphate, and some type of energy. Like the main ATPase that we're going to be concerned with later is called the myosin ATPase. That's really what is breaking it down to have all of our cross-bridge cycling and things. But that's not important yet, but it will be. Now here, the structure of ATP. I want to guide your eyes to right here, to right there, the amount of squiggles. And this is going to be important for effectively what we can even take off of this ATP. So I think all of us know we can take one of them off, right? So ATP, take off one of the phosphates, and we get some energy, and now it's an ADP, and like an inorganic phosphate, and we got some energy out of it, right? So the amount of squiggles here, one squiggle there, that essentially notates that it can be taken off. So if we can take one of them off through that whole ATPase reaction, what happens whenever we take the second one off? And I want you to think about this in regard to endurance signaling. So whenever we break down ATP, we're left with ADP. Now, if we break down even more ATP, we can get another ADP. And that will leave us with that equation. So we can do this, right? So ADP plus ADP yields ATP and then AMP. So what I want you to know is the enzyme here is called meaning muscle, myokinase. So everyone go and write that down. There's also another one that is like that's not in the muscle called denylate kinase, but I'm not concerned about that because I care about muscles in here. So here, that myokinase is the enzyme that functionally can make it to where ADP plus ADP gets us more energy. Now, someone tell me what the endurance signaling cascade is. Thank you, Matt. AMPK, PGC1 alpha, and then we get more like mitochondrial biogenesis, all of that type of stuff. What stimulates AMPK is this AMP. That goes and stimulates AMPK, and then that leads to like more mitochondria and everything. So building on stuff that we already talked about, I want you to like understand that this is functionally how endurance is even being signaled, like how we get more mitochondria, how we get better at burning fat, all of that. It's due to a deficit of ATP, and we get a bunch of AMPs, and that stimulates AMPK, and then mitochondrial biogenesis happens after that, right? So like that's how I'm trying to like tie this into genetics. Everyone good with this so far? Good? Okay, cool, good. Like here, it's a picture from your book. I think it looks pretty. Pretty much this is just the relationship between like endergonic, exergonic, anabolic, catabolic reactions. Almost always we're like breaking down ATP to like fuel something within the cell. Neither here nor there. But next, going into like some bioenergetic stuff, let's see what this even says. There's effectively two pathways how we can make ATP. So there's the anaerobic pathways and the aerobic pathways, like with those like last two bullets there. So anaerobic, I think everyone understands, it does not involve oxygen, right? And that is functionally two different things. Like one thing that we did in lab, like whenever we were talking about like phosphocreatine and that whole pathway, we're going to talk about that here. And then there's the aerobic pathways collectively, like more or less like the Krebs cycle, electron transport chain, and just virtually called oxidative phosphorylation. So all of those terms are kind of grouped together in these ways. What else? Phosphocreatine breakdown, glucose and glycogen. So degradation of glucose and glycogen both being called glycolysis. So like it just kind of tie up some of these bullet point loose ends. Good. All right, next slide we're going to talk about phosphocreatine. Any questions on this? So just collectively anaerobic and aerobic pathways going together. Some of these pathways you don't need to breathe, other ones you do. However, breathing is good. It's consistent with life. So good. We need to do this. Okay. This next pathway, anaerobic ATP production, really there's two that are notated here. ATP-PC system, also called the phosphagen system, also called the PCR system, whatever we want to call it there. This is where creatine is a big deal. So within the cell, we have phosphocreatine, which this is where we get one of those phosphates from, or where we can. So whenever we have an ADP, phosphocreatine can donate its inorganic phosphate to the ADP, and now we have ATP and a free creatine in there. So there's one way to do it. We can also do it through this whole myokinase pathway, which the myokinase pathway is really more related to like endurance signaling. This one up here is more related to like kind of strength signaling. And we'll talk about supplementation with it just here in a bit. But glycolysis, effectively, there's an energy investment phase and an energy generation phase. So overall, glucose, whenever it's aerobic, leads to 2 pyruvates or 2 pyruvic acids. It would technically be called pyruvic acid if it's within a muscle cell. If it ever got exported out of the cell, it would be called pyruvate. Or 2 lactic acids if it's anaerobic. So right here, go and just memorize these points, and we're going to look at where all this happens. So energy investment phase, it requires 2 ATP. So we have to donate 2 to get energy out of it. You have to spend money to make money, effectively, is the analogy that's always used in this case. The energy generation phase, this produces 4 ATP, 2 NADHs, and 2 pyruvates or 2 lactates. So all of that really good, important, memorizable, quizzable next time information. That was a hint. Right? Okay. Okay. Now, does creatine supplementation improve exercise performance? Who's heard of taking creatine to be better at stuff and things? Right? Hopefully everybody. It is one of the most studied substances on the planet, and at least in regard to supplements, ergogenic aids, improving performance, and things like that. And I actually know almost all of the people that started all of this research. So like Darren Willoughby out at Baylor University, Rick Kreider out of Texas A&M. I think he was at Baylor whenever he was doing this, though. But really wonderful guys, great scientists. Effectively, what comes of this? If we have more creatine, we can have more creatine phosphate within the cell. Everyone good with this so far? I think that makes sense. So like you can get phosphorus or phosphate from just about anything, like drinking stuff, eating things, whatever. Like that doesn't really seem to be much of a limiter. But if we take creatine, we can have more phosphocreatine or creatine phosphate within a cell. That means we can replenish more of these ADPs more quickly. So overall, what it does, it seems to extend how long you can have high power output. So here's a question that always comes up, and it's probably going to come up on the test, so just be ready for it. Does creatine increase maximal or limit strength? What do we think? Like a 1 repetition max bench press. If I give you creatine all week, is it going to increase it? Probably not. In fact, there's been studies on this, and actually my mentor Mike Roberts, him and I, we had a bet on it, and I won because y'all don't care about what I do, so I'm not going to talk about this. But effectively, what it will do, once you reach sufficient saturation of creatine within a muscle, it will take a set to where if you could do, say, 5 repetitions with a given weight or maintain power output for like 10 seconds, now you can maintain that power output for 12, 13 seconds, or do 6 or 7 repetitions. That's effectively what creatine does. Now, if we have a lot of creatine inside of your cell all of the time, and now you can always do 2 extra repetitions or have a high power output for an extra like 3 seconds, do you think that will lead to adaptation over time to where you are actually stronger? Bingo is correct. Shaking his head, yes, of course it will. Like, hey, whatever you're doing 3 sets of 10 with right now, imagine that 3 sets of 12 feels exactly the same. And then you continually do that, and then over time your biceps are bigger, and then spring break's all great for you, right? Because like that's, I don't know, like the 16-ounce curls, you never get tired doing them. I'm trying today. Okay. Now here, something that, like those of you who are kind of in the know, there's a couple of different flavors of creatine. Like there's creatine ethyl ester and like some other ones that like people at like say GNC might want to sell you. Now the only one that actually seems to be effective at all and is the cheapest is actually creatine monohydrate, meaning that it's creatine bound to like a single water molecule, and it's the cheapest. Like I don't know, like you could buy a year's supply for like a couple of dollars or something like that. Now, like it's probably like closer to $50. I'm not sure what you think, Josh, but it's Okay, cool. So 72 servings, that's, like a certain amount of time. Right, good, perfect. Like I was gonna say how many months that was, but I can't do math in front of you right now, so. All right, cool. Next thing. Moving on, this is just kind of one of the blurb boxes. Any questions about creatine, really quick? No? Now, there actually is a little bit of research in like endurance athletes who take creatine. Like anybody who's like ever ran a race, like you kick at the end, right? Everyone knows of this, right? Like the last, I don't know, mile, 800 meters, whatever it is, wherever you kick. Individuals who take creatine, like endurance athletes, they do kick a little bit better, but there might be a little bit of a trade-off to where they might weigh a little bit more, so they might get a little bit more fatigued. So there is a little bit of water retention associated with like taking creatine. So it's kind of like a balance there. Yes, Zach. Oh yeah. No, like there was a kid who had like liver damage from it, but like whenever they went back and actually like investigated this kid was doing all sorts of other stuff. He was never drinking water. It's like how many of y'all have been in like middle school or high school? Right? Right? All humans in here, I assume, right? How many of you actually like drank enough water, got enough sleep, took care of your body, all that type of stuff, right? So like what Zach is bringing up, there was a particular individual, and like mothers across the nation are really worried that if you take creatine or protein supplements or whatever, that's going to damage your kidneys, your liver, or what have you. Almost never. Well, in fact, I would say never. I would bet my career on it, the little I have still. Yeah, it's almost like the vaping and e-cigarette stuff, which just about anybody who does the e-cigarettes and vaping and all that. Now, like I understand there's some talk about like the vitamin E being in there, like if you get like vitamin E, like alpha tocopherol in your lungs, that's bad for you. But almost any of the time, like the people I know that vape or whatever, they do cocaine more than regular people. They don't sleep as much. There's so many confounding variables that like it's virtually not worth us talking about. Are you all good with that? Right? So like whenever they were talking about that like vaping e-cigarette thing, my numbers might be wrong, but there's around 50 people that die in pools every day. Let's ban pools, right? More people die of that, or more people die from vending machines falling on them or whatever. So like, which we probably should ban vending machines, but that's neither here nor there. Yeah, that worked out well for Bloomberg in New York, but whatever. So here, lactic acid or lactate. So here's just, it's a memorizable thing. Lactic acid, this is what it's called inside of the muscle. Lactate, this is what it's called inside the blood. So whenever we were measuring it in the lab, in people's blood, we were actually measuring lactate. Now, like whenever it's inside of your muscle, it's called lactic acid. Just a particular note that now, like if you use them interchangeably in conversation, that's perfectly fine, whatever. But like just on the test, I want you to use these correctly. Everyone good with that? Right? So nomenclature and things. And like actually, I don't know, just a little bit of an aside, so y'all like know where I'm coming from. This guy, Bruce Gladden, they call him Dr. Lactate, and he actually is at Auburn, where like I got my PhD, and like he's been called Dr. Lactate for a very long period of time because he's, as it says right here, he's among like the 3 leading people like in the universe about lactate, and he was my biochemistry teacher, and yeah. I was actually the only kid that made an A in that class. Now, I think it's because Gladden and I got along well. I actually still email him every birthday that he has. January 27th, I believe, is his birthday. But it's That has nothing to do with class. Okay, cool. Moving back. It's I miss Gladden a lot. He's a good man. Okay, so the two phases. Like here, we already talked about this a little bit. Just here's a pictorial representation of it. So the energy investment phase, essentially 2 ATP required. Then the energy generation phase. Particular note, like knowing really well the 4 ATP and 2 NADHs. We really need to know that. Really need to know that. Okay, going ahead. Now, within glycolysis, the whole 10 steps of it, I've always gone back and forth in my head about making y'all memorize every single step, every enzyme in it. And I'm not going to. But I'm going to make you memorize all the steps where something interesting happens. So we're going to be talking about a lot of enzymes. There's a lot of stuff to memorize from here going forward. All right. So this first thing, how we get glucose into the cell. You need to know what enzyme does that. And it's called hexokinase. Now, it's called hexokinase in the muscle. In the liver, it's called glucokinase. But in here, again, only care about muscle because biceps. So hexokinase. This is where that first, like ATP, is given off. So blood glucose, we need to put a phosphate on glucose so that it can't get out of the cell. It's frequently called the sugar trap step. So essentially, it traps it to where like now it has to stay in the cell. Because like we don't have the enzyme that actually takes off that phosphate inside of muscles. So anytime we get glucose into the cell, it has two fates. It's either going to be burned or stored as glycogen. Like it can't come out. All right, so that's the first step that we need to know about. And here, the glycogen, this glycogenolysis, it's called glycogen phosphorylase, and we'll talk about that more in exercise metabolism. But note right here that it doesn't cost us an ATP. That's going to be important later. Right now, it's not, but it will be soon. So hexokinase, getting blood glucose to glucose 6-phosphate inside the cell. It can't leave, and now it has one of two fates. Glycogen or has to be burned. Okay, now energy investment phase. A couple of more places. So this top one right here, phosphorylation of glucose by ATP produces an activated molecule. So right there, that's hexokinase. We don't need to look back at that. So we have that phosphate on there. Notice C6, that's how many carbons, and that P notates how many phosphates are on it. So the next one that we need to know is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. So what that is called is PFK, or phosphofructokinase. So know this, that it converts fructose 6-phosphate to fructose 1,6-bisphosphate. And we lose an ATP, converting it to ADP, and that's where that phosphate goes, into the double phosphated, like 6-carbon molecule right there. Now, those of you who struggle studying with these things, I would spend a crap load of time with this. A bunch. Because like if you haven't taken biology classes, or many of them, this seems like a different language, right? But you need to know this. If you don't understand how this works, I don't care about like what you say about like deadlifting or bench pressing or getting your biceps bigger. If you don't understand how energy works within the system, this is really important. Good. Okay. PFK. And like I actually found this. It's so this is out of like Quebec. It's so they speak French in Quebec. So PFK. All right. Yes, Sydney. Yes. It is technically the rate-limiting enzyme in glycolysis. So this is probably the most important enzyme in glycolysis. And it adds a phosphate from ATP to fructose 6-phosphate to yield fructose 1,6-bisphosphate. Everyone good with that? Right. Now, here, moving on. Those are the two ATPs that we lost. right? So energy investment phase, over. Now let's start getting stuff out of this, right? And Kentucky Fried Chicken, which, pi pollo, right? You know, so French, Romance languages. It works. Okay. All right, here. Now, there's a particular enzyme that gets fructose 1,6 bisphosphate. It cleaves it into 2 different 3-carbon molecules. I don't care if you know what that enzyme is, so saving you some time. Just on this energy generation phase, know that there's 2 branches of it. That one side is 3 carbons, the other side is 3 carbons, and we're interested with what's going on there. Now, glyceraldehyde 3-phosphate. This is where I really wanted you to remember what dehydrogenases do and what kinases do. Because if I give you these steps, you should be able to kind of guess what the name of the enzyme is. So dehydrogenase, that does something with taking off a hydrogen, right? Removing a hydrogen. Hopefully everyone remembers that. So glyceraldehyde 3-phosphate dehydrogenase. Do y'all see how that's easy? Right? So to kind of let the cat out of the bag a little bit, I might give you this on a test and be like, hey, where are the important enzymes and spell them? Right? Okay. So here, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. That is a dehydrogenase enzyme. Now, how do we know that it's a dehydrogenase enzyme? It's removing a hydrogen from glyceraldehyde 3-phosphate and putting it on NADH. So here is where the 2 NADHs are coming from. That's going to be important in the electron transport chain. Everyone good with this so far?
Where did the glyceraldehyde 3-phosphate come from?
Oh, it's this enzyme called like aldolase. It converts fructose 1,6 bisphosphate. Yeah.
Glyceraldehyde 3-phosphate.
Yeah. Enzyme that I am not super concerned with y'all knowing about. So good question, though. Good question. All right. So here at this reaction, what I want you to know, glyceraldehyde 3-phosphate dehydrogenase removes a hydrogen from glyceraldehyde 3-phosphate, and there is more or less a free-floating phosphate. It's actually from like a triose, but like that doesn't matter. We get a phosphate from somewhere and we add it to the system. So we take off a hydrogen and we find a free phosphate and add it to the system. And that's how we get 1,3-bisphosphoglycerate. Notice there that there's 2 phosphates on it. So up here, glyceraldehyde 3-phosphate, 1 phosphate. Here afterward, it's still a 3-carbon molecule with 2 phosphates on it. Everyone good with that? Right? And phosphates, as y'all can tell, are important, right? So good. Next thing. Hopefully, it makes sense that this reaction is going to be really important for us. So 1,3-bisphosphoglycerate, 2,3-phosphoglycerate. We get an ATP from there. So we're going to take off one of those phosphates with this enzyme, phosphoglycerate kinase. So that phosphoglycerate kinase, we're essentially adding a phosphate from 1,3-bisphosphoglycerate to an ADP molecule to get an ATP. So now we're making money. And notice that we made 2 ATPs here. Everyone good with this so far? I promise I'm almost done with class today. The next one, that's important. Phosphonenal pyruvate to pyruvate. We get an ATP there. 2 more, one on both sides. Pyruvate kinase. Hopefully this one makes sense. Like down here, pyruvate kinase, we're taking that last phosphate off, and we're left with a 3-carbon pyruvate. Those are all of the enzymes I want you to know, bless you, in glycolysis. Next time, we're going to move into the Krebs cycle and the rest of oxidative phosphorylation. Everyone good with this? Any questions about anything? Okay, cool. Rock on. See you tomorrow.